the mitochondria. As we can see from our TPT, the mitochondria is a double membrane organelle with a matrix and his own ribosome and the mitochondrial DNA. So, and the mitochondria also has the ATP synthesis. So the most important function of mitochondria is to produce energy through the process of oxidative phosphorylation. That is the most energy that we use to maintain our movement and some for our brain to work or our muscle to move. So it is the common type of the energy is the ATP. And the mitochondria also has some other function. For example, regulate the metabolic activity of the cell. Or it will also promote the growth of new cells and cell multiplication. The mitochondria also plays an important role in apoptosis or programmed cell death. And the mitochondria also can help maintain an adequate concentration of calcium ions within the compartments of the cell. So, the mitochondria have very important role in our cell, and it is located on the cytoplasm of a cell. So, if and also the mitochondria has his own DNA. So, if the DNA in the mitochondria mutate, that this mutation will cause some phenotype in human body. Today we will learn about this aspect. First, we will learn about the mitochondrial genome. The mitochondrial DNA, also known as the human chromosome 25. Do you know why it named the chromosome 25? We know that we humans have 22 pairs urochromosome and with X and Y chromosome. Total is 24 chromosomes. So the mitochondria known as the chromosome 25. It is an organelle outside of the nucleus containing the genetic information. Its genetic characteristics are non-Mendelian inheritance. Because when a cell divided into cells, the mitochondria is randomly enters two daughter cells. So it is non-Mendelian inheritance. And in 1981, Anderson completed the sequence of human mitochondrial genome. At that time, He said the human mitochondrial DNA has 16,569 base pair. But in 1999, Cambridge University sequenced the mitochondrial DNA again. Finally, they get the conclusion that the human mitochondrial DNA has 16,568 base pair. So here from our PPT, you can see the human mitochondrial DNA has 16,568 base pairs. Next, we will see the composition of the mitochondrial genome. The mitochondrial genomes are a neat closed-loop double-stranded DNA molecule with 16,568 base pairs, and it is a closed-loop double-stranded DNA molecule like a plasmid of a bacteria. The mitochondrial DNA, including a heavy strand and a light strand, and the heavy chain is rich in guanine, and the light chain is rich in cytosine because they are complexed to each other. It is a double-stranded DNA strand. And the G pairs with C. The guanine pair with a cytosine. And the mitochondrial DNA can be divided into coding regions and no coding regions. As we can, you have learned about the DNA of the nuclear in human. The nuclear DNA also has coding region and no coding region. But the mitochondrial DNA have some difference with the nuclear because there is no intron between the coding region and the no coding region. The genes in the coding region are arranged very compact, and some region, maybe some region overlap. And there are no promoters and introns, no termination codon. It usually ends with a urine and adenine base with U or UA. And the no-coding region, also named as the D lobe. Here, from the picture, we can see here is a D lobe. It is about 1,122 base pairs, and it contains the heavy chain replication start point and the heavy chain, the L chain promoters here. Named as pH and PL, promoter for heavy chain and promoter for L light chain, and also the origin of the heavy chain. And the D lobe is a conserved sequence in the mitochondrial DNA. And the coding gene, the coding region is also a conserved sequence, and 75% of nucleotides are homologous among different germlines, including 37 genes. In the 37 genes, 2 genes encode the mitochondrial ribosomes, ribosome RNA, the 16S and 12S. Here you can see the mitochondrial ribosome RNA has the subunit of it is 16S. It is the same, identical with the prokaryote ribosome RNA. And 22 genes encode the transport RNA in mitochondria. And the last 13 genes encode the protein associated with the mitochondrial oxidative phosphoric acid, the OXPHOS system. It is the main function of the mitochondria. So here we can see in the mitochondrial DNA, only 13 genes encode the protein of the OXPHOS system. Here you can see the complex 1 about 7 genes, and complex 3 1 genes, and 3 genes for complex 4, and 2 genes for complex 5. So most actually, the human mitochondria has about 1,500 proteins, but only 13 proteins are encoded by the mitochondria. So more than 1,000 proteins are encoded by the nucleus and then transported to the mitochondria to carry on their function. So here we see that the mitochondrial genome inheritance is semi-autonomous. So the human mitochondria has about 1,500 proteins. Please close your microphone. 王美花 please close your microphone. Okay, let's continue. So most of the protein subunits and other proteins, because here we can see the human mitochondria has about 1,500 proteins, but the mitochondria DNA only encodes 13 proteins. So most of the protein subunits and other proteins that maintain the structure and function of the mitochondria depend on the nuclear DNA, the nDNA, and they are synthesized in the cytoplasm and transported into the mitochondria by specific transport mode. So the expression of the mitochondrial DNA gene is restricted by the nuclear DNA. And the assembly and maintenance of mitochondrial oxidative phosphate system requires the coordination of nuclear DNA and mitochondrial DNA. They work together to participate in metabolic regulation of the body. So, the mitochondria is controlled by two genetic systems, the mitochondrial genome and the nuclear genome. So the mitochondrial genome is semi-autonomous. Because the mitochondria has its own DNA. The DNA needs to replicate and transportation and translocation and transcription into messenger RNA and then translocate into amino acid. So, how does mitochondrial DNA replicate? The mitochondrial DNA can also carry out semi-reserved replication, and here it's the starting point of the heavy chain replication, the origin of the further heavy chain, and the L chain replication. Origin, the OL and the OH are separate by about two-thirds mitochondrial DNA. From here, from the picture, we can see here is the origin of the heavy chain, and here is the origin of the light chain. And the direction is clockwise. So we can see the light chain. The origin of the light chain is about two-thirds from the of the mitochondrial DNA from the origin for the heavy chain. And the replication starts from the transcription promoter of the L chain in the control region. First, a piece of RNA is synthesized using a L chain as a template as the primer for the heavy chain replication. Then, under the action of DNA polymerase, is the primer synthesized by the L chain as a template. Then the heavy chain starts to replicate. And under the action of the DNA polymerase, a complementary heavy chain is synthesized to replace the parental heavy chain and complete to the light chain. Here, the red chain is the new heavy chain, and the substitute heavy chain of the parent remains in a single-strand state. Here. And the region where the substitution takes place is called the substitution ring or the D ring because it looks like the character D. So it also named the D ring. So this type of DNA replication is called the D ring replication. And when the heavy chain achieves the new heavy chain achieves the origin for the light chain. So the light chain starts to replicate, but its direction is opposite to the heavy chain. So it is a counterclockwise direction. But now the heavy chain has already synthesized about two-thirds 1/3 of the mtDNA. So when the heavy chain finish the replication, the light chain only in the length of the light chain is only 1/3 of the mitochondrial DNA length. This picture is also shows the uh, mitochondrial DNA replication type, the D replication. So here we should found the D replication. Finally, after the heavy chain and the light chain finish the replication, then two new mitochondrial DNA appears. Then they will start another replication cycle. The mitochondrial DNA can replicate, and it also needs to express in the mitochondria. So next we will learn the transcription of the mitochondrial DNA. Compared with the nuclear gene transcription, the mitochondrial DNA transcription has the following characteristics. First, both chains of the mitochondrial DNA have encoding functions. Because the nuclear gene only have one strand can be as a template chain to synthesize messenger RNA, but both chains of the mitochondrial DNA have the encoding function, so they can work as a template to synthesize RNA. And both strands are similarly transcribed from the promoter of the D region at the same rate, but the L strand transcribed clockwise and the H strand is counterclockwise. Here you should pay attention that the transcription of the L and H strand, the direction of the transcription of heavy and light strand is opposite to the replication direction of the mitochondrial DNA. Because during the replication, the heavy chain is clockwise and the light chain is counterclockwise. But during the transcription, the F strand is clockwise and the heavy chain counterclockwise. And also the mitochondrial DNA has no terminators between genes. And here you should another character of mitochondrial DNA. The tRNA genes are usually located between mesRNA genes and the ribosome RNA genes. Here, from the picture of the genome of mitochondria, you can see here is the gray is the ribosome RNA genes, and the blue region is the genes encoding protein, and the black line is the tRNA genes. So the tRNA genes also located between messenger RNA genes and ribosome RNA genes. And the most of the genetic code, the triplet codon, is same to the nuclear DNA, but the mitochondrial DNA also has his own genetic code. For example, here, the AGA codon. The AGA in the nuclear, it is the ARG amino acid, but in the mitochondria, it is the stop codon. And the AUA in the nuclear, it is the ILE amino acid, but in the mitochondria, it is the MET amino acid. And here, the CUA codon. In the nuclear, it is LEU amino acid, but in the mitochondria, it is THR amino acid. And here in the UGA, it is a stop codon in the nuclear gene, but in the mitochondria, it is a TRP amino acid. It can translate to amino acid. And the TRA in the mitochondria is highly versatile. Here, the codon, the triplet codon for the VLA valine amino acid is GCU, GCC, GCA, and GCG. And oh, sorry. The codon for the here is the codon for the here's the codon for the ALA amino acid, aligning amino acid is the GCU, GCC, GCA, and GCG. And the anticodon in the nucleic tRNA is GGC and UGC. But in the mitochondrial DNA, only one anticodon is enough, is the UGC. So the tRNA in the mitochondria is highly versatile. Yes, because the mitochondria has its own DNA, and the DNA during the replication, it also will occur mutation. So next we will see mutations in the mitochondrial genes. Since the discovery of the first mitochondrial DNA mutation in 1988, more than 100 disease-related point mutations and more than 200 deletions and rearrangements have been identified. And among them, about 20%, 60% point mutation affect transport RNA, and 35% affect subunits of polypeptide chain, and only 5% affect the ribosome RNA. Because here you can see only 2 genes encode the ribosome RNA, and 22 genes encode the tRNA, and 13 genes encode the polypeptide chain. So most of the mutation will affect tRNA genes, and major percent will affect polypeptide chain, and lesser percent affect the ribosome RNA. The mutation rate in the mitochondrial DNA. The mitochondrial DNA mutation rate is 10 to 20 times higher than nuclear DNA. Why? Why this phenomenon? Why is the mitochondrial DNA mutation rate higher? There are some reasons. First, the mitochondrial DNA has a very compact array of genes, and any mutation of it may affect an important function region of its genome. And the second, the mitochondrial DNA is a nicked molecule. It will not bind to histone, so it lacks of histone protection. And third, the mitochondrial DNA is located near the linear membrane of mitochondria and is easily damaged by uh, oxidative damage due to direct exposure to superoxide particles generated by respiration. By respiratory chain metabolism and hydroxyl radicals generated by the electron transfer. First, the mitochondrial DNA replication frequency is high and the replication is asymmetric. Fifth, the mitochondrial DNA lack a genome, lack of effective DNA damage repair ability. So, due to these 5 reasons, the mitochondrial DNA mutation rate is higher than nuclear DNA. And here, there are several criteria for determining whether a mtDNA mutation is a genetic mutation. It is also work for the nuclear gene mutation. One, the mutation occurred in a highly conserved sequence or at a site of significant functional importance. Two, the mutation can cause respiratory chain defect, so is the mutation affect the function of the mitochondria. 3. The mitochondrial DNA mutation type was not found in the normal population, but the same mutation was found in patients from different families with similar phenotypes. So, if you want to determine the mutation is the genetic mutation for a certain disease, then you need more than 2 families with similar phenotypes, similar disease to study to carry on your study. 4. There were impurities, and the degree of the impurity was observatively correlated with the severity of the disease. So here, we learn impurities. What is impurity? We will learn about it later. So there is 4 criteria for determining mitochondrial DNA. Whether a mitochondrial DNA mutation is a genetic mutation. And the mutation type of the mitochondrial DNA, because in the nuclear gene mutation, it has a dynamic mutation, and stable mutation. And in the stable mutation has point mutation, fragment mutation, frame shift mutation. And in the point mutation, we have this what types of the mitochondrial DNA mutation. The mitochondrial DNA mutation types mainly include point mutation, large fragment recombination, and the mitochondrial DNA quantity reduction. So, the mitochondrial DNA mutation has three types. First, the point mutation. Most of the point mutation is missense mutation. Two-thirds occurs in genes encoding tRNA and ribosome RNA, and one-third of the point mutation occur in the gene that encodes the messenger RNA. The related disease and the MERF, it affects the tRNA gene. Also, a famous one of the mitochondrial DNA, the LHON syndrome. It affects the messenger RNA for the ND4 complex. It is a labor hereditary optic neuropathy. And also another is the KSS. It is the KSS type mitochondrial encephalopathy. And here, the mitochondria. For you guys, you should think which organ in our body needs the mitochondria or needs the energy most? You know? Hello? So, which organ in our body needs the energy most? Is the brain and the muscle. The brain needs the energy to think, and the muscle needs the energy to move. Yes. So, the mitochondrial DNA mutation mostly affects the brain or the muscle, and if it affects the brain, it named the mitochondrial encephalopathy, and if it affects the muscle, it named the mitochondrial myopathy. Next type is the large fragment recombination. It contains includes the deletion and repetition. The deletion of large fragments often involves multi-genes, which can lead to the decline of mitochondrial function and the reduction of ATP production, thus affecting the function of the tissues and organs. Some common deletion is from 80,483 to 13,459 base pair. For example, the KSS ischemic heart disease, and in the 8,137 to 13,033 disappears. It is a degenerative disease associated with aging. And another common deletion is the 4,300 and 389 to 14,000 and 1812. Most affect the energy metabolism is destroyed. Last is the reduction of mitochondrial DNA amount. The decrease in the amount of mitochondrial DNA can be autosomal dominant or recessive, indicating that the disease is caused by nuclear gene defect caused by mitochondrial dysfunction. Because there are mutations of the mitochondrial DNA, then it will also have the mitochondrial DNA damage repair. There are two main mitochondrial DNA repair mechanisms. One is a excised repair. The other one is a transfer repair. In the excised repair, the endonuclease excises the damaged DNA fragment first, and then DNA polymerase copies the correct nucleotide sequence to fill the vacancy formed by using the undamaged strand as a template. And in the transfer repair, the transferase recognizes the mutant nucleotide, such as a methylated nucleotide, and then removes the mutant nucleotide. So here is why the mutation rate in the mitochondrial DNA mutation rate is high, because it is lack of effective DNA damage repair ability. We see the mitochondrial DNA genome. The mitochondrial genome is a semi-autonomous because it is controlled by the nuclear genome and the mitochondrial DNA. So, in here, in our today's class, we most talk about the mutation of the mitochondrial DNA because in the mitochondrial DNA mutation, it will affect the some function of the mitochondrial DNA, and then because the DNA is in the mitochondria, then it will affect his offspring by a non-mendelian inheritance type. But we also know that most of the functional proteins that need the nuclear DNA to synthesize. So it's the mutation of the nuclear encoding mitochondrial protein have mutation. What will happen? First, it will have the defect in the electron transport chain, and then it will maybe a defective mitochondrial protein transport, maybe the defective substrate transport. All these three aspects will affect the function of the mitochondria. But here, because the mutation in the nuclear gene that encoding mitochondrial protein, so the genetic type of this is the Mendelian inheritance type. Finally, we will see the genetic characteristics of the mitochondrial disease. The first famous character is maternal inheritance. In Chinese, it's named 母系遗传, the maternal inheritance. Because during a oxide formed, the sperm and the egg fusion, but the sperm mostly often provide a DNA to the egg cells. So most of the cytoplasm, nearly the whole cytoplasm, is the egg cell. So the mitochondria is in the cytoplasm. So the mitochondria of oxide is from his mother. So it is a maternal inheritance. It means the mother passes the mitochondrial DNA to her sons and daughters, but only the daughters can pass her mitochondrial DNA to the next generation. Another one is the humour plasmid and the heteroplasmid. In there are several criteria for determining whether a mitochondrial DNA mutation. The first one is the impurities. Here is the homoplasmy and the heteroplasmy. If the mitochondrial DNA molecules in the same tissue or cell are identical, they are said to be homoplasmic. In cloning and sequence studies, it has been found that some individuals have two or more types of mitochondrial DNA at the same time. This is called the heteroplasmy. The mechanism of this, the heteroplasmy, also named as the impurity. The mechanism of the impurity occurrence may be that the mitochondrial DNA mutation leads to the existence of both wild type mitochondrial DNA and mutant mitochondrial DNA in a cell, or the heterozygous mitochondrial RNA existing in the zygote is randomly assigned to the daughter cells during cleavage. The mitochondrial DNA impurities also differ in different tissues derived from this differentiation. And the rate and the incidence of impurities in different tissues are different. Because we have done that, I talked about them before. The brain and the muscle need more energy, so it had more mitochondrial quantity. The incidence of impurities in central nervous systems and muscle is higher, and the incidence of impurities in blood is lower. The incidence in adults is much higher than that in children, and the incidence of impurities increases with age. In heterotype cells, Y-type mitochondrial DNA protects and compensates for mutant DNA. So, the mitochondrial DNA mutation does not have immediate serious consequences until it reaches a certain dose. Then it will have the disease phenotype. In here we will learn about a bottleneck effect. In fact, the bottleneck effect is only in genetics, but in other conditions. But here in the genetic, the bottleneck effect means each human egg contains about 100,000 mitochondrial DNA, most of which is lost as the oocyte matures, and the number can rapidly drop to less than 100. The process in which the amount of the mitochondrial DNA decreases dramatically during egg formation is called genetic bottleneck. So here from the picture we can see the egg contains many mitochondrial DNA, but during its maturation, most of the mitochondrial DNA is lost, and only a few mitochondrial DNA left. Then this mitochondrial DNA pass through the oocyte then to his offspring, to his children. So this phenomenon and the mitochondria pass through the next generation rapidly. So the offspring of one mother has different types of mitochondrial DNA. The third one is the third characteristics of the genetic character of mitochondrial disease. One is the maternal inheritance. Two is the homeoplasmy and the heteroplasmy. Third is the thread effect. The mitochondrial DNA mutation can affect the function of mitochondrial OSPOS system and cause ATP synthesis disorder, leading to the occurrence of disease. But in fact, the relationship between genotype and phenotype is not so simple. The expression of mutant mitochondrial DNA is affected by the impurity level of mitochondria in cells and the minimum energy required to maintain normal functions of tissues and organs, resulting in different penetrance and expression. Only when the mutant mitochondrial DNA reach the minimum thread, then the disease will occur. If the mitochondrial DNA is less, it will not cause the disease phenotype. So the phenotype of the impurity cell depends on the relative ratio of mitochondrial DNA mutations and wild-type mitochondrial DNA in the cell, and the minimum amount of mitochondrial DNA mutants that can cause dysfunction of a particular tissue and organ is called the thread. The thread is a relative concept that is susceptible to changes in mutation type, tissue and aging degree, and varies greatly among individuals. So the thread is common to the organ that needs the energy most or less, more or less. So it is different in tissues and aging degree and varies greatly among individuals. And the mutant mitochondrial DNA accumulates gradually in cells with age, so mitochondrial disease often presents as an age-related progressive exacerbation. For example, in a family with the MERF disease with broken red fibers, 80% of the mitochondrial DNA mutant individuals have mild symptoms at age 20, but were clinically severe at the age 60. So the heteroplasmy with low penetrance of mutant mitochondria below thread, it has the cell or the organ or the mitochondria has normal function. And if the heteroplasma with thread percentage of mutant DNA, it will have abnormal function. And if it is a high percentage of mutant DNA, it will abnormal function, sometimes severe disease or alter the phenotype. So here, have only very little heteroplasm because the white type mitochondria can compensate the mutant function. So the mitochondria has normal function. Why this phenomenon occurs? Till now, few studies for it. So this need you guys to work study to learn about this phenomenon. The last characteristic of the mitochondrial disease is the unequal mitotic separation. It means during the cell division, the mutant mitochondrial DNA and the wild-type mutant DNA are separated and rapidly assigned to the daughter cells, so that the daughter cells have different properties of the mutant mitochondrial DNA molecules. This random allocation leads to the process of mitochondrial DNA impurity changes called the replication separation. Here, during the cell division, the mitochondrial random segregation to his daughter cells. Here is all the wild-type mitochondria. Here has two the heteroplasmy about 30%, and the heteroplasmy is 70%, and it is all mutant homoplasmy. This phenomenon called replication separation of the mitochondria. So last, we have a summary for today's class. Maybe today's class you can summarize that the mitochondrial disease inheritance is easier than single gene disorder or polygenetic disorder, even than the population disorder. So today's class, you should grasp the characteristics of mitochondrial genetic systems. And during these characteristics, there are some other knowledge points for you to grasp. One is a semi-autonomy. Why it is a semi-autonomy, you should know. And it has different genetic codes. Most of his genetic code is the same with the nuclear, but he has some own genetic code. 3 is a high mitochondrial DNA mutation rate. Here you should know why the mitochondrial mutation rate is higher than the nuclear DNA mutation. The fourth is maternal inheritance. Why the DNA is a maternal inheritance? The fifth is the femoplasmy and the heteroplasmy. Here we learn the bottleneck effect. What is a bottleneck effect and what is the homoplasmy and the heteroplasmy? This is the thread effect. What is a thread? And why the thread effect occurs? Because the Y-type mitochondria can compensate for the mutated mitochondrial DNA, so it is a thread effect. And the next is the replication separation. So what the replication separation will bring to mitochondria? You should also know. Okay, here is all conditions for today's class. Do you have any question?
没有问题，老师。
Okay. Today's class is over.
OK，老师，Thank you，老师。
Thank you，老师。
Thank you，老师。
再见，老师，谢谢。